Assalamu alaikum wa rahmatullahi wa barakatuh. I think most of you already got tired after these three sessions. Thank you very much for the organizing committee, especially Dr. Prof of Immunology and Dr. Mansoura. And it's my pleasure to be here to give this brief talk. And actually, this is the third time to be at Mansoura University to give talks on immunology, and the second time to be on this conference. The first session was rich in clinical application of stem cell therapy. The second one was actually rich of some mix of clinical and preclinical application. And this session, I start with a little bit different talk about stem cells. So everybody today is talking about how to get benefit from stem cells or how to differentiate stem cells into adult cells. I think this is the opposite here, which we're trying to reprogram adult cells, specifically T cells, to become or at least acquire stem cell phenotype. And this was something, one of our focus in the last years, and I'll show you some of our data generated from our lab. Before, three topics. The first one is how to activate or to harness immune cells, either at the level of innate or adaptive immune cells. And we were able to do that. The second one is how to understand how cancer cells escape from immune cells. And we were able to focus on two very important cells. They are regulatory T cells and myeloid-derived suppressor cells. And we have several publications on this in leukemia, in lung cancer, in breast cancer, and also in preclinical models. The third one, recently we started at Tanta University at Cancer Center of Excellence in Cancer Research, DMARC. It's a CCR, Center of Excellence in Cancer Research. Most of you know, in clinic, of course, the cancer therapy is based on surgery or radiosurgery, and this is, of course, effective but does not impact on doesn't clear the metastatic cells and also does not induce memory cells against cancer. We are interested actually in immunotherapy, and in clinical trials there is some trials now and which reach in the clinic. Most of them right now is based on antibody-based immunotherapy, and the Nobel Prize this year was awarded to two scientists, one from US, one which block the function of immune cells and enhance the growth of T cells, of cancer cells. The other one is cytokine-based therapy, and toll-like receptor-based therapy, and of course vaccination. What we think is that adaptive cell therapy is very important, and we focus on this topic, whatever it's T-cell based therapy or dendritic cell based therapy. And we have done some work on NK cells, but it's a little bit different story. So I focus today on T-cell based therapy, specifically CD8, which is the target cells for cancer cells. And also, as concept-wise, we believe that not only one therapy can eradicate tumor, we need to have combination of all of these, especially cell-based therapy with cytokine-based therapy and toll-like with vaccination. Adaptive cell therapy can be based on adaptive T cells, which are CD8, CD4, LAG cells, lymphocyte-activated killer cells, or innate-based therapy, of course, dendritic cells, which is one of our focus, and in clinic, of course, progenitor cell therapy. And we found, based on our studies, that the cell-based therapy doesn't work perfectly unless if you treat or if you precondition the host with chemotherapy before adaptive T cell therapy. That's why the combination is very important. And here, in case of you, when you apply adaptive T cell therapy, you do leukapheresis for patient, you take peripheral blood, and then expand cells in vitro for a couple of weeks. This is a standard protocol. And when you activate the cells, you infuse them back to the same patient. It's autologous cell-based therapy. With the main goal of these cells transfer the cells to home to a tumor microenvironment, attack tumor and became tumor cells. This is the best scenario we are looking for with adaptive cell therapy. Actually, in the IH group and the other groups, and we have done also several studies on adaptive T cell therapy, we found some challenge. The three main challenges is the long-term activation to generate sufficient cells, and by the end of 3 weeks or 4 weeks, we found most of cells die, even though they are very active, but they die quickly when you infuse them back to the same patient. The second one is short persistence of adaptively transferred T cells. Even the cells which survive and traffic into tumor, they die later on. They don't persist for a long time. The third one challenge is poor survival of the transferred T cells. When you take these cells ex vivo and measure the cytolytic activity, you found them they are already exhausted. So they cannot form memory cells. How can you overcome these three challenges? Actually, this is a typical scenario of how you generate anti-tumor immunity. This is the tumor cells. They shape tumor antigen into the tumor microenvironment or even integrate plasma, different tissues in skin or whatever into lymph node. So they need some factors which are inflammatory cytokines which activate immature dendritic cells to mature dendritic cells. Mature dendritic cells, they are opposite to immature. They are poor at uptake, processing, and killing, but they are very good. They are excellent at presentation of tumor antigen by MHC+1 or 2 to CD4 and CD8 T cells. When this occurs, of course, if we are lucky enough, the CD8 cells by the help of CD4 can attack the tumor and kill it. However, this doesn't happen when you have large tumor or even at early stage. So one strategy is to work on how to manipulate dendritic cells to make it more mature, and this can be, as we have done this several times in several publications, we take the blood, take monocytes from blood, culture them in, for 7 or 8 days, and generate the immature dendritic cells, and of course add some inflammatory factors like 1 or 2 of these to generate dendritic. This is one approach, and we have been successful to do so. And these cells, after generation, we add the tumor antigen in vitro before vaccination. This is one approach. I'm not going to talk about this today. The other approach is to how to manipulate CD8 cells, especially CD8 cells, to be more effective to attack specifically the tumor cells. Actually, taking in consideration these two pathways, we focused on, we decided to work on 3 pathways or 3 concepts. The first concept of generation of T cells in vitro and modify them in a different way to make them more active and more capable of killing a tumor. We use a different approach using IL-12. In most of the previous studies in NIH and other groups, they used IL-2, IL-7, IL-15. Considering these cytokines enhances survival and proliferation of T cells, but we used IL-12, and I'll show you fantastic data about IL-12. The second approach is to work on the tumor, how to condition the, how to get benefit of chemotherapy, short-term chemotherapy, in this case, CTX is cyclophosphamide. And we understood the cellular and molecular mechanism underlying the effect of chemotherapy. I'll show you, and actually, no, I'll show you some of our publications. The third one is how to get this microenvironment after chemotherapy to be very active and create very stimulatory microenvironment. And this also we have worked in this and been very successful in our publication. I focus on this approach today about how to use IL-12. And we use, we started with animal models. There is very nice animal models, transgenic animal models. In these models, they are genetically engineered CD8 cells. Are genetically engineered to specifically recognize some specific tumor antigen, in this case melanoma. We use this just as a model. So you have donor transgenic mouse and the recipient transgenic mice. They are different in one allele, which is LY5.1. They are isogenic, similar in everything except LY5.1 and LY5.2. So we transform CD8 T cells, which can only recognize but cannot attack or kill the tumor cells, only recognize. And we transfer very few number, almost 1 million or 1.5 million into recipient, and then track them in peripheral blood or spleen, and you can very nicely identify donor cells, CD8 cells from host cells. This one. So this is the transfer of transgenic cells, and we can, of course, characterize the phenotypes very nicely by flow cytometry. We use this very nice model, and we published several papers about the second concept I told you about, which is preconditioning the host before adoptive T cell therapy with chemotherapy. The chemotherapy here we did a series of studies, and we come into several mechanisms, but very interesting one which I show here is the expansion of the dendritic cells after almost 12 days, from 10 to 12 days after chemotherapy. But in nature, and we were able to turn them into natural dendritic cells with full IC, which is full like receptor 3 agonist. And then we come into another series of publication, the kinetics of rebounding of lymphoid and myeloid cells, and then the dynamic alteration and the molecular mechanism, and we determined the very important role of the LIT3 ligand, which actually mobilize stem cells from bone marrow to peripheral blood. And then we use the toll-like receptor, as I mentioned to you, to enhance the tumor microenvironment, and then we published one very nice article that cited many times. In this article review, based on our studies, we defined the two, which already known, the lymphopenic phase after the treatment with chemotherapy, in this case cyclophosphamide. Lymphopenic phase and the recovery phase. Of course, these two phases are known, but what which hasn't been known is what happened in each phase, especially at the cellular and molecular level, and how we can get benefit from each phase. So at the lymphocytic phase, for example, we can use TLR agonist, or like receptor agonist with vaccination, and then similar way at certain time points at the recovery phase. Using this dissecting model, very nice model, we were able to eradicate melanoma using adoptive cell therapy of naive CD8 cells and vaccination with peptide, in addition to bolo-IC, which is toll-like receptor 3 agonist. However, we started to think about if we can omit vaccination and just use the cells alone after we condition them with IL-12 without vaccination. And this actually came to our mind because we have another story about IL-12. It started in 2001, and technical issue here. some story about IL-12 in 2001, and we were able to treat tumor-beating mice with IL-12. At that time, IL-12 was a very hot topic, and I guess either Roche or another company, they were marketing IL-12 for clinical trials as a single treatment to eradicate metastatic tumor. But they found, unfortunately, significant toxicity. They stopped, and they even lost a lot of money after they manufactured a lot of quantities of IL-12. So a company called the Marine Polymer Biotechnologies, they came to us and asked if we can package IL-12 in nanoscale of nanogel, and we were able actually to generate very, very significant anti-tumor response using IL-12 as compared to systemic IL-12. And this was published in Journal of Immunology a couple of years ago, almost like in 2004, almost like 14 years right now. And we were very enthusiastic about IL-12, and we tried to apply for clinical trial, but nobody was enthusiastic about that because at that time I told you IL-12 was a drawback. So we wrote a review article about that issue to summarize our work, and came into a conclusion that to everybody, IL-12 can be very good if we can package it in a smart way in nano gel. But we got disappointed, and we started to think about how to come back with IL-12 in a different way. And this is, we started our story, and we used adoptive cell therapy of CD8 cells using the same animal model I told you about, but without vaccination. And we published a series of studies, very nice studies, about 7 or 8 studies. And all of these studies showed the capability of IL-12 to condition CD8 T cells in vitro very nicely, just in 3 days. In MIH group, they use 3 weeks of IL-2 culture in vitro, but we used only 3 days, and we were able to program CD8 cells to show better survival, better trafficking, better capability of killing cells, and so on in these studies. And we, at that time, we didn't know why IL-12 is doing so. Why IL-12 not IL-2 or IL-7 or IL-15, even though they are very effective cytokines. And we did one paper actually by MIH group. Rosenberg, it's a very famous guy, Ristifo, and Rosenberg, he's the guy of adoptive cell therapy, who is the one who started with the theory of adoptive cell therapy. And they have plenty of papers about it. They know everything, either in mouse models or in human, about CD8 cells, the pros and the cons of this. But they showed in this paper in mouse models that hematopoietic stem cells promote the expansion and the function adaptively transferred onto tumor CD8 T cells. What they found is that you don't have to use chemotherapy to condition the host before therapy. Simply, you can co-administer CD8 T cells, bone marrow CD8 cells in this case, with CD8 T cells, and you get the same effect as the combination of chemo and the CD8 T cells. So I actually stopped at this paper, and I started to think, can we turn CD8 cells to acquire, or maybe IL-12, actually, this is what came into our mind, maybe IL-12 enhance or reprogram CD8 T cells to acquire stem cell phenotype. This is what we know about the differentiation of stem cells into myeloid progenitors, ending with neutrophil, monocytes, and other cells, platelets, and other cells. And the other pathway end with dendritic cells, natural killer cells, lymphocytes, either B cells or T cells. This is known. So, is it possible to take T cells back into the phenotype of hematopoietic stem cells. This was our question using female mouse model. I told you already about this model is based on transferring of melanoma-specific CD8 T cells from donor into recipient, and then with or plus cyclophosphamide, just single dose, mild dose, and then adaptive transfer of CD8 T cells. This model actually mimic the human melanoma very nice. And these CD8 cells we use, they can recognize GB100, which is a kind of peptide expressed by melanocytes. So using this model, we take CD8 cells, culture them simply just for 3 or I guess maximum 7 days in culture, and then we called day 3. And this was the first time to culture cells only for 3 days. And the reason we thought about that is when cells are, and this is based on the Restifo NIH global study, found when you culture cells for a long time, they got exhausted. So they are not good enough. So we decided just to culture for 3 days or for 7 days, and then with or without IL-12. This is sham. PMEL is the name of CD8 transgenic CD8 cells because the mouse is called PMEL. And we culture cells and get on by forward side scatter, and then with sham and IL-12, and then looked at the percentage of CD8 cells. Most of these cells, after 3 days even, are CD8 PMEL cells, and other cells die, most of them. We looked at 3 kinds of characteristic feature of any CD8 cells. You need to use them for adaptive cell therapy. We used the activation phenotype, we're using CD69 and CD25. We used the functional phenotype based on production of granzyme, which cytolytic mediator, which attack cancer cells or target cells, and interferon gamma as well. And we used also proliferation capacity in vitro, can proliferate much better than the sham. And we also characterize the memory phenotype. So the 4 features, I guess, of CD8 T cells. This is in vitro. These 4, starting with activation phenotype of day 3 only of female IL-12. And using these 2 markers, we found female IL-12 as compared to sham, or to 7 IL-15, or combination even of 7 and 15, they show much better early activation marker. Both of these markers are called early activation marker because later on they downregulate, their expression is downregulated. The same, when we looked at the granzyme and the interferon gamma, which are the cytolytic mediators of CD8 T cells, we found also the interferon gamma, especially interferon gamma, is expressed much nicely better than the other groups. The same with proliferation with IL-12 at day 3. So taking this and also memory, we used 2 markers, 2 markers for early memory cells and another 2 markers for central and effector memory phenotype of CD8 cells, and these are known, established to characterize the efficacy of anti-tumor efficacy of CD8 cells. And in our case, especially with CD127, we found IL-12 also is much better. Then we came into the question: does these cells, as compared to those cells as compared to sham, they show anti-tumor effect? And this figure show you, this is the mice bearing melanoma treated with BBS, and this line treated with CTX. They show some actually delay of tumor, but the tumor come back. And then female sham, the same thing, as because also it got cyclophosphamide, and this one is IL-12. As you can see, up to 35 days, there is no tumor, which was surprising for us. And this data actually already published, so there is no wanted to show you. And then we compared the IL-12, this one, with other cytokines, which have, of course, 2, 7, 15, and the combination, and we got the same. These cytokines conditioned cells, yes, they can delay, but the tumor come back, I guess, at 40 days or something. So they show immediate anti-tumor effect, but the tumor come back. But in case of IL-12, it was amazing. The tumor does not come back at all, at least at this point, 52 days. And this without vaccination, only cells conditioned in vitro with IL-12. And then we looked at, this is just representative data showing you, actually, because we use B-mel model, and the GB100 is expressed by melanocytes. So when you treat cells, okay, so you got vitiligo in here because, of course, they have expressed GB100. So you got this white, and this indication of the cells already moved, acted into tumor site, and killed the tumor. And this was a site of tumor already cleared. However, in case of sham, the tumor is still there, and actually the time we sagged the mice at this time was early, this was like after 3 months or something. So this just to show you, at the solid tumor, you can infuse cells and home into tumor and attack and kill the tumor. And very nicely, without any tumor left. We used another model which is based on liver metastasis. You inject the tumor cells, which is called the B16 melanoma. They are black cells, of course, because they express they are melanocytes, and infuse them through intravenous injections. They home into liver, into blood, not into liver. So we this show you just some lines from control and sham and B-mel island. This was also amazing for us. There is no any tumor in the lung, which confirmed that these cells can move into subcutaneous tumor microenvironment, the primary tumor, and also can come into the metastatic tumor. Then we asked if these cells, how long these cells can survive or persist in vivo. We took several actually organs, blood, lymph nodes, liver, and spleen, of course. We looked at the number of these cells here, and as you can see, the number of cells, of course, decreased by time, but still at day 35 you can detect. And by the way, 3.5 is significant number of cells after a month or so. And then we also looked at these cells, at the kinetic of these cells on day 7, 10, 13, 90, and more days. Here, almost a year, 232 days, and you can see, still you can detect the cells in blood, lymph node, and spleen. And actually, we analyze these cells in blood even after re-challenge of B16 melanoma. So re-challenge of tumor does not negatively impact the existence or persistence of these cells. So somehow the IL-12 does something in these cells which allows them to survive and persist for almost a year in tumor-bearing mice, even if you re-challenge the mice with the tumor. So this is the conclusion of day 3, B-mel IL-12, which we conditioned with IL-12. One is survival, better survival, persistent, longer persistence, proliferation, higher proliferation in shorter time, just 3 days, anti-tumor efficacy against solid or metastatic tumor. So why IL-12? Why is such 3 or 4 parameters occur nicely with IL-12 better than other cytokines? We started to think, as I told you, when we read this paper about the stem cell phenotype by these cells. this was something unusual to think this way, but this after we read the paper of Restifo. And we looked at SCA1, and this is in mice, of course, SCA1 and CD44, and we found these cells with stem cell phenotype express 77% of SCA1 as compared to other, of course, other conditions also expressed, but this was very high as compared to other conditions. Also, we looked at the position of SCA1 and CD44 in spleen. Is it systemic or only blood? We looked at spleen, lymph node, bone marrow, blood, and we found also the same characteristic features of, especially in lymph node. And as you know, lymph node is very important target site for the interaction between CD8 cells, CD4 cells, and the antigen-presenting cells. So this was very nicely exhibited. This is only on female IL-12. This just, and we actually looked at donor and the host. And of course, as you can see, only donor show these cells, host doesn't show a lot, which confirm the in vitro. Then we decided to run some array of real-time PCR on two kinds of genes. One is related to stem cell phenotype, and the other one related to drug-resistant genes. And we found, and we, of course, the ratio between the day 3 IL-12 and the day 3 sham, and we found it's in most genes, especially for stem cells or for drug-resistant genes, they are much higher on IL-12 females than sham. And then when we found these genes related to drug resistance, we sought to harvest cells and look at them in vitro and treat them with dinuroxin, which is almost like doxorubicin, but you can use it in vitro, not in vivo. And we compared the naive, early sham, early female IL-12 in vitro, and we found the number of cells killed by dinuroxin is less than the naive or early. But of course, it's not, especially between sham and IL-12, not huge. But when we turned it into in vivo after 90 days, we found the donor. We did not find female sham, of course. We find only IL-12 female, and the percentage was 20% of donor cells. I'm almost done. Yeah. And this is for me, right? Yeah, I'm almost done. Yeah, that's nice. Yeah. And we thought to test if these cells can negate re-tumor re-challenge. So we challenged the tumor and adopted the transformed cells. And as you can see again, even after re-challenge, the cells are capable of eradicating tumor. So I come into the final 3 slides. This, which I started with, can we, at some level, reprogram adult cells to acquire a stem cell phenotype? This was our initial question. I guess somehow, yes, we can. Of course, this is not easy. You may not induce most of the phenotypes, but at least the phenotypes you need to for better survival, proliferation, trafficking, functionality, and in case of anti-tumor, this is, this slide actually, before I finish my talk, the adaptive T-cell therapy become a fascinating approach in clinical trials. And there is a technology called the CAR, it's chimeric cells, and these cells, you take CD8 cells and transfect them with a TCR. TCR is the molecule in T cells which recognize the cancer cell. So you come into very specific, very effective, very efficient TCR molecule on CD8 surface and transfect into any CD8 cells, naive CD8 T cell. And then grow them in vitro, okay? And then you grow them in vitro and transfect them and activate them and then infuse them back. So the infused cells are naive cells, but they express TCR, which is very strong to recognize and attack tumor cells. And this, and of course, this induced cell death. This, if you write on Google, you will find how CAR cells in clinic now in Europe, in US, and Japan. I'm not sure about China, but it's this is based on the dogma of adaptive T cell surgery. Of course, this requires, it's on shelf right now. You can have the vector with a specific TCR. It's used mainly against B cell acute lymphoblastic leukemia in children and some lymphoma. So they are, it induced 95% clinical response. And this, some companies, of course, FDA approved, and this is based on adaptive T cell surgery. Our approach, because here it's hard to, I don't know about the logistics to import such kind of on-shelf TCR genetically engineered T cell. So I actually worked in hopefully some day to start the clinical trials on IL-12. When I was in US, we started 2 clinical trials and worked very well with IL-12. Hopefully we can reproduce this data in human, as I told you, because it's very simple, it's delayed, and not very complicated, just IL-12, and IL-12 is commercially available FDA. And right now I would like to thank Dr. Sharif Zidane is here, and the other groups here in Egypt and in US, and University of Miami, Medical University of South Carolina. And before I end, just one slide, Dr. Mansoura, I would like to mention about CCR, which is Center of Excellence in Cancer Research. This is in Tanta University, Tanta University Teaching Hospital, 3rd floor, and we have such kind of 4 facilities, and it's our pleasure, of course, to collaborate with in one view. We have 10-color flow cytometry unit. It's 9-color cell sorting unit, isoflux. We have cell culture unit, very nice genomic unit. We have Unix also, like the one Dr. Tanta has, and also we have DNA sequencing, MySeq. Of course, plus the other basic equipments like bioanalyzer, we have PCR, and such kind of thing. And thank you very much for my team in Tanta University. These guys actually are like 1/3 or 1/4 of our team. We have a very nice team, undergraduate, postgraduate, and also sometimes high school students. And thank you very much.
Thank you, Dr. Mohamed, for this excellent talk. Any question? Sorry.
Thank you very much, Dr. Mohamed Labib. As usual, your talk is very impressive and informative. I am interested in CAR-T. When we can do it in Egypt? What are the challenges facing CAR-T?
Yeah, I think the challenge is logistics, not anything else. I mean, how can we do some agreement or import such kind? The car right now is on shelf, so just based on the vector. The transfection doesn't take a long time and is not complicated, just 3 days in vitro and very bone marrow based transplantation lab. You can do it very nicely with someone who is trained well in culture. So, and you can characterize it, the viability, the cells after that, the functionality, no problem. I think it's a logistic, how to convince or to contact who we can contact to get. There are 4 companies actually working on CAR technology. So they started with personalized medicine 4 years ago, but right now, luckily, they have on shelf this vector, which can be transferred without any safety issue. So I think, I'm not sure, I cannot answer this, but maybe the first step is to talk to the company And I have a friend of mine in the United States, he's working on that, and I talked to him. He said maybe the first step is you can send someone, physician, to watch the process from A to Z and watch the patient in injection monitoring, and then you can start talking to the company which supports this.
I think we must do this.
Yeah, hopefully.
Because it's time.
Yeah, it's very powerful, yeah.
Yes.
Very powerful, really.
Okay, we can talk
Yeah, sure.
after that for this issue because I am really very, very impressed by the results.
Yeah.
Okay. Thank you very much.
Which vaccine?
vaccine.
Yeah. Well, it's already, the dendritic cell-based vaccination is a kind of personalized medicine. So because you need to use autologous dendritic cells. But it's already,
يعني الفرق هنا ان احنا استخدمنا
no, IL-12, I used it with CD8, not with dendritic cells. Dendritic cells, we use what's called BOM-IC, which is full-like receptor agonist. I did talk just briefly today. But with CD8, we use IL-12, which is also FDA approved. You can notification.
اشمعنى بقى الفيسكوستين ميلانوما؟
What's that?
الفيسكوستين ميلانوما اشمعنى؟ وهل
No, we used it just as a model, not for any other reason, because this model is very nice. You can judge your data very nicely and precisely. But we started with the dendritic cells. We started with collaboration, and we published this last year, 3 years ago, with a group at Medical University of South Carolina. We treated the pancreatic cancer patient with the dendritic cell-based vaccine. And you can look at this Salem, dendritic cell-based vaccination, and work it very well. With, and this was pancreatic. They also started, like I'm not involved in this phase 3 trial, with colon cancer, and worked it very well. So, but in and the melanoma, common and the melanoma. But in this case, we just use the.
هو الفيسكوستين جين ميوتيشن برضه بتبقى انفولفنج بين الميلانوما والبنكرياس؟ مش عارف بالصدفه ولا ايه؟ هو الفيسكوستين جين ميوتيشن بتبقى انفولفنج البنكرياس والميلانوما مش عارف بالصدفه ولا ايه؟
only lung.
تمام
بيعمل metastasis in the lung.
تمام
في another cells we can talk later on. The homing is cell activated.